Assalamualaikum. Well, hope you're doing well. I'm also fine. Currently it's 10:24. Time to say good night, but before this, I think we should go through a small lecture. And today our lecture is about mitosis. First, we'll see what is mitosis. Today we study about mitosis and its different phases as well. Okay, what is mitosis? It's a process in which cell divides into two daughter cells with the same number of chromosomes. Means the number of chromosomes in the daughter cell, the product of the mitosis, have the same number of chromosomes as like in the parent cell. Okay, what is the importance of mitosis? Well, mitosis is very important. First, it helps in the growth of cell and repair. It helps the cell to divide and produce new cells. Then it also helps in repairing the damaging tissues. The second importance is the renewal of tissue. Cells that are lost or damaged due to injury or normal wear and tear are replaced by the process of mitosis. Asexual reproduction. Different organisms like bacteria, protists, and the plant, they produce asexually, and they produce offspring which are genetically identical to the parents, and these are all because of mitosis. So the first three importance are the growth of the cell, repair of the damaged cells or the tissues, then the renewal of the damaged tissues, then asexual reproduction in different types of organisms. Now come towards the other importance. Here the fourth one is embryonic development. Mitosis is responsible for the rapid cell division, means an abrupt or a very fast division of the cell, due to which it gives rise to a number of other cell types and tissues, which as a result in the development of an organism. In this way, it helps in the development of an embryo. As we know that mitosis is basically occurs in the somatic cell, and we know that the somatic cells are the body cell. That is why it helps in the development of an embryo. And then the wound healing, it helps in the rapid proliferation. Proliferation means the production of cells at the site of injury and helps in the healing process. The last one is maintenance of chromosome number. As in the first slide, we see that in the process of mitosis, what happens? Daughter cells are formed with the same number of chromosomes. In this way, we can say that it maintains the genetic integrity. Means it maintains the number of chromosomes in the offspring or the daughter cell, just like in the parent cell. Now come towards its phases. The first phase is prophase. Here are some bullets which shows the sequence of events that occurs in the first phase of mitosis, which is the prophase. The first one is chromatin condense. Means in the first phase, chromatin condense coiled and formation of highly ordered structure called chromosome. As a result of this condensation, highly ordered structure formed, which are known as chromosome. Then what happens next? The chromosome, they bound together at the centromere. Then kinetochore in the centromere helps the chromosome to attach at the spindle. Kinetochore is a protein which helps the centromere to attach the chromosome at the spindle. Centrioles duplicate. Two daughter centrosomes are formed. Centrosome moves towards the opposite poles. These are the sequence of events which occur step by step in the prophase phase. Then microtubules are formed by tubulin protein. Microtubule, they form spindle fibers. Set of spindle fibers formed, they are known as mitotic spindle. Nuclear envelope and nucleus disappear and then spindle moves towards the center of the cell. Okay, here is a picture. First of all, you people, I think right now seeing these structure, star-like structure, they are basically centriole, which duplicates and moves towards the opposite pole. Centrioles, and these are the chromosome. Okay, here chromosome is in the form of chromatin. Then this chromatin condense and they form chromosomes. And there is a small structure right in the middle of the chromosome, is known as centromere. Inside the centromere there is a kinetochore and one thing more, these are the spindle fibers. Okay, and and this is the interphase, the phase before the prophase, in which the chromosomes are present in the condensed form in the form of chromatin. Then they coiled and they form chromosomes, highly ordered structure, and this phase is known as prophase. Now the next phase is metaphase. What happens in metaphase is growth of spindle fiber at the specific length, kinetochore fiber attach at the kinetochore of chromosome, formation of metaphase plate, non-kinetochore fibers attach with each other. Now come towards the diagram. Here is your metaphase phase, and right now these are the chromosomes. The kinetochore, the center part of the chromosome having centromere in which the kinetochore is present, they attach with the kinetochore of these spindle fiber. Okay, means kinetochores are present in both places, in the centromere and in the spindle fiber. So the kinetochore of the chromosome present in the centromere attach with the kinetochore of the spindle fiber, and they form this metaphase plate, just like in the equator of the cell, the middle of the cell. Okay, this is the metaphase. Anaphase. Kinetochore spindle fiber, they pulls the chromosome. Sister chromatids of chromosome divides and moves towards the centrosome. Then identical copies of chromosomes separate into two groups, means same copy of chromosome, they separate into two groups after the division of sister chromatids when they move towards the centrosome. Okay, and you know what is centrosome? The centrosome, they are basically the division of centriole. These are the centrosome. Okay, and now sister chromatids separate. One move towards this pole, one move towards this pole. Now what is sister chromatid? The sister chromatid basically this part. This and this part is sister chromatid. This and this part is sister chromatid. This and this sister chromatid, sister chromatid. The identical part of chromosome is known as sister chromatids. And this is the anaphase, in which the sister chromatids separate and moves towards the opposite poles. The last phase is telophase, which is basically the reverse of prophase, in which the nuclear envelope comes back and is integrated into in the prophase, and the chromosome, they again unfold and unfold back and they form chromatin structure. Okay, and here is the diagram. See, this is telophase. The both cells, the two daughter cells having the nuclear envelope, which removes in prophase. Again they come back in the last phase, which is the telophase, and both daughter cell have the same number of chromosomes. Thank you so much. End of the lecture. Bye bye.